Okay, so eventually it's trying. So we walked through, basically we got through all of respiration proper on Monday because we kind of finished rounding out the Krebs cycle. We'll spend a little time Friday when we start kind of reviewing all of chapter 7. We should theoretically finish chapter 7 today, and then start the last chapter on Friday. So we'd walked through, all right, this is like the, and I think we're already like halfway. Whatever, that's fine. So these figures are in here. I feel like because they put all the Krebs cycle together like this, it is a little bit overwhelming looking, but don't get too kind of bogged down in this. We'll kind of walk through this in a couple, I've got a couple summary slides we'll do next. But basically kind of be watching the electrons coming off and be watching where we're breaking the carbon bonds, right? Because the whole point of this process is to take this 6-carbon glucose, right, that has energy stored in all of these little carbon bonds, right, to break those bonds, to release energy, and to pull off electrons to use later, right? So that's kind of, I don't need you to memorize enzymes. I don't need you to memorize a bajillion chemical names or anything like that. Just watch, right? We come into, for example, we come into the Krebs cycle with what's left of glucose is 2 little 2-carbon molecules, right? And so each of them come in, we can follow one here, it tacks onto this little oxaloacetate bus or whatever and comes around. We pull an electron and we break one of those 2 carbons off. We pull off another electron and we break the last carbon off. We kind of use what's left in here to make an ATP. We pop off a couple more electrons, right? That is the Krebs cycle, kind of as we need it, right? So we pull off, we pull off electrons 3 times using NAD, 1 time using FAD, and we get 1 ATP out of it, right? Which is kind of the useful parts of the Krebs cycle. So, and this is just kind of the more, if this is helpful, if it's helpful you to see kind of like what the molecules look like, you can use this to kind of reference. If it is not helpful, just keep going, right? So that idea of kind of following the carbons, right? Because that's the point, right? We're breaking these molecules, right? These catabolic reactions to release energy. Yeah. So in glycolysis, where are the carbons going? In glycolysis, we started with glucose, which has 6 carbons, and glycolysis broke it into 2 3-carbon pyruvate molecules. That is what happened, right? We went through that one little kind of, as we were moving into the mitochondria, we oxidized that pyruvate, broke off 1 more carbon. We've got 2 little 2-carbon molecules, right? Those 2 2-carbon molecules go into the Krebs cycle, break off those last 2 2-carbons, right? And now glucose is gone. We have completely pulled glucose apart, broken all of the bonds, pulled off all of the electrons we can. And so glucose has become, right, the products of respiration, 6 carbon dioxide molecules, which you then, there they go, they're gone, right? And then energy in the form of, at this point, 4 ATP, right? We actually made 6, but we had to spend 2. So 4 ATP and 12 electron carriers, right? 10 of those NADHs, each picked up 2 electrons each. And then 2 FADHs, each picked up 2 electrons each. So we are sending our 4 ATP out to do whatever they need to do, and then I can math. 24 electrons are going down to the electron transport chain, which is where we're headed next. So electron transport chain. And this process that is called chemiosmosis, which is where we're actually going to really crank out some ATP. So we're not quite there yet. All right, so let's talk about the electron transport chain first. So, like we just said, from glycolysis and Krebs, we've got 10 NADH to FADH, each with 2 electrons. We've got a couple dozen electrons that we've pulled off of our 1 glucose molecule, and they move to the inner mitochondrial membrane, right? Remember, just briefly, mitochondria has an outer membrane and then an inner membrane. So we're sending all of these guys to the inner membrane, to the electron transport chain, all right? And so what the electron transport chain is a series of 3 membrane-bound proteins with 2 little electron carriers in the space between the proteins, okay? So we've got these three proteins, NAD dehydrogenase, the BC1 complex, and the cytochrome oxidase complex. 1, 2, 3. Between them, we've got ubiquinone and cytochrome. We'll get to them in a second. First, at the NADH dehydrogenase, this is where NADH is going. Not surprising. NADH goes to the NADH dehydrogenase enzyme, that's really hard to say all together, and drops its electrons off here, right? So since we've pulled these electrons off, right, we have oxidized NADH, we now have NAD plus that can go off and do glycolysis again, right, and go back to the beginning and start everything over. Now, FADH is floating around here, kind of in the inner membrane, like in the matrix, right in the middle of the mitochondria. It drops its 2 little electrons here in between. So the electrons from NADH and FADH come and hop on this little electron carrier ubiquinone, right? Right, because in between, inside the membrane, what is it in there? So like that's a very abstract question. What is the nature of the middle of a plasma membrane? That would make it difficult for an electron to just bop around in there. So stretching way back to like chapter 3 and 5. How do plasma membranes form? What are they made of? Lipids. They're made of lipids. And these little lipids form membranes by it's one of those characteristics of water that causes them to behave a certain way. Hydrophobic. Exclusion. Yeah, there it was. Right? And so, right, we've got these polar heads and these nonpolar tails. Can you push an electron through a bunch of nonpolar tails? No. So that's why we've got to have this carrier, right? So we've got these electrons that come into this protein from NADH. We've got electrons that come in and hop onto this carrier from FADH. And so ubiquinone moves it from NADH dehydrogenase, drops these electrons off at the BC1 complex. All right. This all seems silly, right? We're just shuffling things around. We'll get to it. BC1 complex just picks up all of those electrons, passes them on to cytochrome. Cytochrome carries it over to the cytochrome oxidase complex, right? And so what's happening here, right? This is just, we're pulling electrons off, putting them here, pulling them off, putting them here, pulling them off, putting them here, right? We're moving these electrons through these proteins, right? This is just, you remember that kind of staircase of us moving and pulling electrons until we've kind of spent them all out, right? We've moved them once or twice to get to this point. Now we're just like going nuts, pulling them off, putting them on, moving them here, putting them here, pulling them off, moving them here, putting them here. At the end, when it gets to cytochrome oxidase, those electrons are absolutely toast. They're done. They're not good for anything anymore. So this is where we mentioned that idea of the last electron acceptor. This is where that happens, right? So if, like in yourself, if you breathe oxygen and your cells have oxygen available, these little tired, spent out electrons get put on oxygen, right? Along with 2 protons, which means you get water, right? Which is a byproduct of respiration, right? So this electron transport chain, but why on earth is this happening? What good is this doing? Anything at all? Well, what we'll see is that as these electrons are moving, we'll unpack this a little bit more, but these electrons, this charge moving through this membrane and through these proteins is causing these proteins to push protons into the intermembrane space of the mitochondria, right? So you've got the outer membrane, the inner membrane, and like the middle, the matrix of the mitochondria. While this is happening, electrons are getting pushed into the space between the 2 membranes. All right, so we're filling this, what's called the intermembrane space between the inner and the outer membrane with protons. so that's going to matter in a minute. Now, where are the protons coming from? Remember, we looked at this figure, right? I've got my glucose, pops into this enzyme. We're going to oxidize this. We're going to reduce NADH. And when we do that, right, remember we pulled these 2 electrons, we put them on NADH, and we lost, right? They were dehydrogenation reactions. We were popping off protons, right? So those protons have been building up this whole time, right? We've got our glucose minus some electrons, got some protons floating around, and then our electron carrier all filled up. So those protons are coming back into play now. So every time we reduced NAD in respiration, we also got a proton. And so our electron carriers, each with 2 electrons, came in. All these little bopping around protons came in, and we're all in the mitochondria together. So as the electrons are flowing through the electron transport chain, right? Starting here through the membrane, through all these proteins, the charged particles are causing the proteins to move, right? To change shape, right? And they're pump proteins, so their moving causes them to pump protons out into the intermembrane space, right? So we've got stuff here inside the mitochondria, and as we move electrons here, we're pumping protons out into the intermembrane space, which makes a concentration gradient, right? If you push everything to one side of the membrane, you're going to have a high concentration on one side and a low concentration on the other. So we create a proton gradient, a gradient of protons, right? Where they're highly concentrated out here, and there's not many of them in here. All right. So now the matrix, the middle, is going to be negatively charged relative to the intermembrane space, right? Because the intermembrane space is full of protons, which are positively charged. So it's functionally got a nice positive charge in there now, right? So if we've got a positive charge here and a negative charge here, the protons are now attracted to the negative charge, right? That's how charges work, right? Opposite charges attract each other. So the protons are attracted to the matrix, right? They want to get back into the middle. And it just so happens there's an enzyme for that, right? ATP synthase. All right, so if this is that matrix, right, that kind of inner space, and we have the electron transport chain pushing all these protons out here, these protons, right, want to diffuse down their concentration gradient from where there's a lot of them to where there's not a lot of them. And there's this nice little channel here in ATP synthase that will let the protons diffuse back across the membrane. Right, and this enzyme, and you may notice it's going to do something weird because it looks weird, right? Every other enzyme we've drawn has just been a little blob. This looks like some weird, somebody making balloon animals kind of lost their mind. So the movement here of charged particles, right? This thing is actually like a little turbine engine. As you push these charged particles through, the whole thing spins, right? It's like a little turbine. So the protons are powering the spinning pump. And once ATP synthase starts spinning, right, it starts cranking out ATP, all the ADP, right, used up ATP and phosphates that are hanging around, starts generating loads of ATP because it's being powered by these protons that are going through it, right? And so we catalyze the reaction, right, of turning ADP and phosphate into ATP. This process is called chemiosmosis. Oxidative phosphorylation, I think, from like several slides ago. So we're making lots and lots of ATP. 28 to be exact, right? We've been cranking out like 1 or 2 at a time. At the end of that, from that one glucose molecule by using all these electrons, right, that we pulled out of glycolysis, that we pulled out of pyruvate, that we pulled out from those last little carbons left in the citric acid cycle, all of those electrons pass through these proteins, make a gradient, and power this pump that will then, again, using the electron power from this one glycolysis, create 28 ATP. Right. It's kind of our final output. Right. And if we want to kind of pick apart where everything comes from, right, remember we netted 2 ATP in glycolysis, right? We made 2 more in the citric acid cycle, right? And then all of these electron carriers, right, kind of on average, so you got 2 NADH, 4 electrons, get you 5 ATP. In pyruvate oxidation, we filled up 2 more NADH, got 5 more ATP. In the citric acid cycle, we actually loaded up 6 NADH, which gets us 15 ATP. It's not exact, it's actually fractions, but I don't care about fractions, so this is good enough. We're just going to round it up. And then we did have those 2 kind of little slightly weaker FADH. So 2 FADH only gets you 3 ATP, but ultimately you end up with 32 as your net yield. Right?
Net yield is going to be a big thing when it comes to further units. Or
like after unit 3?
Or like after this unit, like, is it going to be something we?
It's something you should know. I'm just saying, like, I just say net yield because technically we made 34. It's just being precise with my words. Not that like, right, we net 32 because we made 34. That's 9. I don't know what that even, we made 34, but we had to spend 2 in glycolysis, so we get 32. Now, just to kind of add one more layer of annoyance and complication. In eukaryotes, we actually only net 30. Because our cells have membranes. And so to get these NADH that we made, these 2 NADH into the mitochondria, we got to spend a couple ATP to get them into the membrane, right? But in like prokaryotes that don't have membranes that they have to pass things through, they don't need to worry about that. So in us, we net 30 ATP total, right? So for every glucose molecule your body breaks down, it makes 30 ATP, basically. All right, so there are a lot of regulatory pathways in aerobic respiration. We're only going to mention a couple, right? So there's lots of different ways, right? There's lines everywhere here, things that are stopping things from happening. We'll give like 2 examples. That'll be good enough for our purposes, right? And it's going to use feedback inhibition, which is handy because we already talked about it, right? When we talked about kind of enzymatic or biological pathways, right? You've got lots of reactions to make something that you need. And when you have enough, right, feedback inhibition is where that end product is actually an allosteric inhibitor, right? It stops an enzyme kind of early on in the process, basically shuts off its own conveyor belt there, right? When you have enough of it. So in aerobic respiration, we see this in glycolysis and Krebs cycle, 2 little feedback inhibition examples. One, ATP is an allosteric inhibitor of phosphofructokinase. Not that you need to know phosphofructokinase, but it's an enzyme in glycolysis, right? So ATP is an allosteric inhibitor of one of the enzymes in glycolysis, right? So when you have enough ATP, it'll actually turn glycolysis off, basically. And then the same thing, right? Because we're also cranking out loads of NADH. And if your cell has enough NADH stored up, it is actually an allosteric inhibitor of pyruvate dehydrogenase, which is in that one little kind of 1-step pyruvate oxidation situation, right? So same thing, if your cell has made enough NADH, it's a feedback inhibitor that will stop respiration before the Krebs cycle, right? So your body has these ways of kind of moderating how much respiration it's doing depending on whether or not it needs to, right? Be processing and spending and harvesting energy. All right. Now, we've walked through all of this kind of on the assumption that we've got oxygen available, right? Because we breathe oxygen and we're kind of using ourselves as an example. But not all organisms, and not even all of your own tissues all of the time, have sufficient oxygen available. So what do cells do when there's not oxygen to put those last electrons on? They do a couple different things. So they either do anaerobic respiration or fermentation. So, right, we've got these tired electrons, and either at the end of the electron transport chain, we just put them onto some inorganic molecule that's not oxygen, right? We'll look at a couple examples. So it's not that much different, but basically the waste product becomes different, right? Instead of having water as a waste product, you have some other chemical that's produced. Or you have organisms that And we'll kind of look at how they truncate respiration. They kind of stop respiration really early on and put their electrons onto an organic molecule. Right? We'll look at both of these. So anaerobic respiration. What if we go through all of respiration all the way to the electron transport chain, and then at the end take that tired electron and just put it on a not oxygen, right? So we can do a couple things. We can oxidize our carbon dioxide, right? Our waste product carbon dioxide, and reduce methane, right? So these are methanogens. So the bacteria that live in cows' guts that help them break down all the cellulose and stuff that a cow can't actually digest on its own, right? A cow's gut does not have a great deal of oxygen in it. So those bacteria have to do something different. And so they just take their tired electrons, they go into methane, which is why cows just belch and release methane pretty much constantly because of those bacteria and the waste product of their metabolism. Conversely, you've got other bacteria in things like, if you've ever been to like Lassen National Park or any of these kind of crazy acidic springs, that will use sulfur instead, and they'll actually release sulfuric acids, right, as their waste product. Because it makes sense, right? Because sulfur, right, we were using oxygen because it was really highly electronegative. So is sulfur, right? It's still in the top right end of the periodic table. It's still a really electronegative thing. It's still fine picking up those tired old electrons. So we can have anaerobic respiration where we use another inorganic molecule to dispose of all our tired electrons. So option one. Option two is fermentation. And fermentation ends up looking more different because if your cells are doing fermentation, your cells are only doing glycolysis. They're not doing any of the respiration that's happening in the mitochondria. They kind of stop short, right? So what happens, right? We're doing glycolysis. We have either don't have or have run out of oxygen, right? But we still need to be recycling NADH back to NAD, right? So we need that for glycolysis to work. So when NADH comes out of glycolysis, it just attaches its electron to some organic molecule, goes back into glycolysis. So it's just a much shorter cycle, right? Frequently pyruvate or something made from pyruvate, right? Because pyruvate is the product of glycolysis. So it's there, it's available. If you do glycolysis, you've got NADH and pyruvate. NADH drops its electrons off on the pyruvate and goes back up, does glycolysis again. All right, so that's fermentation, right? An organic molecule and NADH gives you some reduced organic molecule and NAD that's then available to keep doing glycolysis, right? Usually some organic acid or alcohol, right? Kind of the example that most people are aware of is yeast, right? Yeast is producing alcohol. This is how we produce alcohols. It's also how we make bread. In animals, you see this in the form of lactic acid. Let's look at yeast first. So yeast do what's called ethanol fermentation. So, right, glycolysis happens, makes pyruvate. They go ahead and decarboxylate pyruvate, right? So pyruvate's a 3-carbon molecule. They go ahead and crack off one more carbon. They've got this little 2-carbon molecule. And they use this 2-carbon molecule to oxidate NADH, right? So that NADH drops its electrons off, goes back to glycolysis. And so this is what produces ethanol. This is how you produce alcohol with fermentation, right? And so this production of CO2, right, when pyruvate gets decarboxylated and it releases CO2 gas, this is what makes bread rise, this release of this gas, right? Now, the kind of ironic twist to this process is that, so yeast are producing ethanol as a byproduct. It's a waste product, and it is toxic to the yeast. So normally, kind of out in the environment, right, the ethanol would be released kind of into the environment, not a problem. For people who say homebrewing or kind of doing naturally fermented wines in a closed system, you cannot create a naturally fermented wine with more than 12% alcohol because at that point you've killed all your yeast will all start to die. There's too much alcohol in the wine. There's obviously there's ways to get around that. People make alcohol with much more than 12% alcohol in it, but in kind of a natural closed system, you'll actually kill off all your yeast at 12%. All right, so ethanol fermentation. Option 2, lactic acid fermentation. This happens in animals, this happens in us, right? If we find ourselves lacking in oxygen. So in lactic acid fermentation, we don't decarboxylate pyruvate. We do glycolysis, we make pyruvate, and we just use that pyruvate to oxidize NADH. So we just go straight through, right? And so the waste product here is lactate or lactic acid, right? So if you are doing things where you're using up the oxygen stores in your cells, this happens particularly in muscle cells. Your muscle cells will start to go through this form of lactic acid fermentation because they don't have enough oxygen, which is generally fine because your blood circulating will remove the excess lactate and you will be all right. Now, if you accumulate a lot of lactic acid in your muscles, right, if you work out and you kind of work out past this point, right, your cells become very oxygen depleted. You build up a bunch of that lactic acid in your muscles, and that's why your muscles fatigue. That's why they kind of burn the next day. They are kind of literally full of acid, actually, right? And so sore muscles after a workout like that is a result of a buildup of this acid because your cells at some point in your workout were actually kind of oxygen depleted. Yeah. So metabolism in the absence of oxygen. Check. Last section of chapter 7. We don't just eat glucose molecules, right? You're eating other stuff. So how do other things that we consume get kind of incorporated into this process, right? Let's look at that. So we'll look at proteins and lipids. Most of the other stuff that you're eating that's not some kind of carbohydrate, right? Is it in some way, probably mostly protein or lipid. And it's actually not that tricky when you kind of look at them this way, right? So here, so proteins, right? If you unfold a protein, you spread it all out into its primary structure, it's just a chain of amino acids. Your body can break a bunch of amino acids apart, and an amino acid, like glutamate or any of the amino acids, is just, right, some kind of hydrocarbon, like, right, organic molecule with an amine group on it. So we break our protein into amino acids, we pop off this amine group, right, and the amino group is just waste, like urea. So this, right, is removed from your body in the form of urine. And all that you're left with, right, is this carbon chain. This your body can deal with, right? We can kind of shuffle around carbon chains, make it look like a glucose or a pyruvate or something, right? And then break it down in either glycolysis or the Krebs cycle, right? We can make it fit into the processes we've already talked about at that point, right? Lipids are even easier, right? Because lipids, like the triglycerides we talked about, are just glycerols with hydrocarbon chains attached. So if you break the glycerol off, you just have a bunch of hydrocarbon chains, and it's the same thing, right? You can shuffle those hydrocarbon chains around and then send them down to the Krebs cycle, and the Krebs cycle can handle it, right? Break those carbons apart, pull the energy out, pull the electrons off, and carry on. One thing you'll note, we talked about this before, right? These lipids actually will yield more energy than something like a carbohydrate. They hold more energy. For example, right, we've got this 6-carbon glucose that we've been talking about in respiration. A 6-carbon fatty acid will actually yield on average 20% more energy than, right, a similar carbon chain from a sugar, which makes sense if you just feel like, just kind of feel your stomach. Think about eating, right? Like if you eat a really carby meal and then go work out, it's going to kind of burn off a lot quicker, right? It's not going to last as long compared to, say, if you consume a meal that's higher in lipids or proteins or something kind of heavier that's holding more energy like that, right? So it kind of makes sense even at the very kind of whole body level. All right. This is a complicated chart that you don't need to memorize. It's just to make a point, right? So we have all of these things we're consuming, right? Proteins, saccharides, right? Sugars, fats, all of these things. Sugars are easy, right? They kind of fall straight into glycolysis, pyruvate oxidation, citric acid cycle, blah, right? Lipids, we can break off that glycerol and send those chains, right? Turn them into acetyl-CoA, basically, and into the citric acid cycle. Same thing with proteins, right? Either we can turn them into pyruvate or we can send them onto the citric acid cycle. The idea being that basically your body can basically turn anything you eat either into pyruvate or acetyl-CoA and kind of feed it into the process, right? It's kind of how all of your food comes into this metabolic process. Right, it kind of links everything up with all of these different food molecules that we eat. All right. One last point to make, right, with all of these kind of intermediates, all these things you're taking in, all of these things you're breaking down to harvest energy. When we talked about chapter 6, kind of energetics, how chemical reactions work, how enzymes work, all of this stuff, right, we mentioned that reactions can go forwards and backwards, right, depending on how much reactant you have, depending on all these kinds of different factors, right? And so you'll see this when you look at these metabolic processes, right? The enzymes and the mechanics that your body is using, right, to break down glucose, to break down lipids, work the other way, right? So every reaction you have that breaks down glucose, right, your body can turn that around and build glucose, right? When you need to store lipids, your body can take that process that we just discussed that breaks down lipids. It can kind of switch directions with those enzymes and build lipids, right? Everything in here kind of goes, and it shows in, right, all these arrows go both directions, right? You have these reverse pathways that'll use the same enzymes and just run the reactions the opposite way, right? So for example, glycolysis. Most of glycolysis will run forward like we just looked at it, or backwards, depending on what your body's doing, what it has and what it needs. Yeah. Okay, and I think, yes, that is the end of chapter 7. So I'm going to let us go a pinch early. We're going to start Friday. So take a little time if you have it to kind of look back over chapter 7. We'll open up Friday with a little bit of chapter 7 review, and then we'll start the last chapter of the unit, which will be photosynthesis. Okay. So have a good rest of your Wednesday. And I will see you on Friday.